After completing previous two videos about epithelia and connective tissue, today we're going to address the muscle tissue, and then later we will be also presenting the nervous tissue so that we complete four basic tissue types that the human body is composed of. Let me share the screen and let's go into first slide. Essentially, the muscle tissue is the one which created its own ability to produce contraction. Contraction will result in a shortening of a muscle cell or muscle fiber. We interchangeably use terms muscle cell or muscle fiber, where the word fiber is perhaps just slightly bit better to use because it kind of subconsciously implies the length of a cell. And in this case, some of the muscle fibers could be pretty much of unimaginable length, exceeding 10, 15, 20 centimeters in length, so comparable to about 8 to 10 inches, if not even longer. It would be impossible to start this presentation after introducing ability of muscles to contract, not to immediately address that inside a human body, we have three different types of muscle tissue. The one which is pretty much immediately synonym for muscle tissue are obviously our skeletal muscles, part of our locomotor system, muscles that we voluntarily control, inducing their contraction, and also voluntarily stopping contracting a muscle or a group of muscles, depending what is our intentions. But from histology or physiology point of view, it is also very important to address that these skeletal muscle fibers are also known as striped or striated. We can see stripes or striations on both photomicrograph of skeletal muscle, but we can also see it even more clearly in this handmade diagram illustrating only a section of the muscle cell or muscle fiber. Perhaps another thing that immediately strikes an eye is that we have multiple nuclei that are pushed more peripherally within a muscle cell, and perhaps that would be another surprising moment for us that we'll try to resolve in the next couple of minutes. Why muscle cells would be characteristically recognized by presence of multiple nuclei, because we know it's not a typical thing for human cell to have more than one nucleus, but here in the muscle fibers, particularly skeletal muscle fibers, it is going to be almost like a standard or a norm. Another question one has to answer is, where are these beautifully looking light and dark stripes coming from? Physiology of muscle and its sliding filaments theory, which explains the contractility of the muscle tissue, will give us much more clear answer. But for now, we can say that presence of contractile proteins that are running within the muscle cell following its longest axis, they're having very regimented and very strict overlapping pattern, where obviously where more proteins overlap, they create a little bit of a darker stripe, and where the overlapping area is less visible, basically appearance of a lighter colored band would be observed on the surface of the cell, even when using just a regular light-based microscope. Clearly, this is something that has a very particular arrangement, and we will see with more details what actin and myosin filaments look like, what is the way they overlap, and what generates this kind of very unique pattern, which is typically present on a skeletal or striped muscle tissue, but also we're going to find it on muscle tissue, the second type, which is only to be seen within human heart. For that reason, we're saying these are cardiac muscle cells or cardiac muscle fibers. Now we can see that pattern of stripes alternating light and dark bands are also present, but there is one huge difference that one can see when comparing cardiac muscle to skeletal muscle. Look at this. It appears that skeletal muscle runs practically just one direction, but if you immediately switch to next slide, you realize that cardiac muscle cells appear to be branching. And this is one of the differences that we can see. Stripes are again consequence of the same arrangement of actin and myosin contractile proteins within a cell, but idea of cells branching in the cardiac muscle tissue is something that really supports quite nicely physiology of the heart itself. If we just think that somewhere above or below this diagram, there is a center which is producing some kind of electrical current. Let's put a little electrical sign here and say that is the source of the electrical change. We can now say it's not just a source, but rather it's the cardiac pacemaker. The electricity will spread from a source into neighboring adjacent cells. And of course, the electricity will not be picky in saying, oh, I want to go this way or I want to go this way. Instead, it will spread through any kind of electrical conductors. As we can see here, and the label very nicely points it out, between two adjacent myocardial muscle cells, we have connection, and it is called the intercalated disc. If you remember bonding between cells and connexons that were used as the last example, where cytoplasms of two adjacent cells are in a direct contact, electrical current will pass through the intercalated discs through connexons, and the same electrical event will now start affecting other cell in line and will keep going all around the heart until each and every muscle fiber becomes properly stimulated to do its contraction, which would lead into complete cardiac systole, cardiac contraction aimed to expel the blood from heart's chambers. When it comes to how do we control it, we made it very clear that skeletal muscle fibers are controlled voluntarily. And can we make the same statement about cardiac muscle tissue? Of course, we cannot. We cannot, by our will, change the heart and tell it, okay, now you contract, now you don't, or do a couple of contractions, now take a couple of seconds rest. That is not within our reach. The heart is creating its own pace, and that is what will be explained in cardiovascular system physiology. There would be a section of myocardial specialized tissue which would be generating signals on its own at the pace between 90 to 100 signals per minute, and that will be our sinoatrial node, or what is colloquially referred to the pacemaker of the heart. Should it be left unattended, our heart, regardless who we are, what we do, will keep steadily beating between 90 to 100 beats per minute, but then with the help of autonomic nervous system, sympathetic and parasympathetic divisions, basically they would be able to alter the heart's function. Sympathetic division of autonomic nervous system will stimulate the heart to increase number of beats and also to increase the force of its contractility. So it will result in heartbeat which goes over 100, 120, 130, 140 beats per minute, while under the influence of parasympathetic division of autonomic nervous system, the heart function will slow down, as we should experience in all of us that our heartbeat at rest generally falls in between 60 to 70 beats per minute. So that kind of range that the heart as the principal pump of the cardiovascular system could achieve under control of two divisions of autonomic nervous system basically enables us to operate under different conditions, going from rest to some moments when we have to put absolute and maximal physical performance into our body, which must be supported by cardiovascular and respiratory system at the same time. Otherwise, we would fail in that attempt. So for both smooth cardiac and skeletal or striped muscle fibers. Unfortunately, they have stepped out of cell's predictable cycle and they entered their G0 phase a long time ago. Unfortunately, I say because the damage done to a muscle, whether it's skeletal or cardiac, is not going to be easy to cover by simply asking adjacent muscles to keep multiplying and to replace damaged or torn muscle fibers by new ones and to fully and completely restore the function, whether it's of a skeletal muscle or the heart itself. For skeletal muscles, it might be a little easier to compensate the damage because even if the muscle is completely torn, basically the tear part will heal, but forming adhesion between two different parts of the muscle. That adhesive part will become a scar. It would lose its ability to contract, but at least it will keep the muscle and maintain to a certain percent its original action and activity, whether it's to 50%, 70%, or maybe 80 or 90%, it is irrelevant for this point. Heart muscle, unfortunately, suffers a damage, which is most typical scenario when a person suffers from a heart attack. Basically, insufficient blood supply to myocardial cells are going to lead to death of certain part of the myocardium. And depending on how extensive or how minimal the damage is, that is going to determine the outcome, where some people, unfortunately, are unable to survive massive heart attack, which affected a large area of the heart, denying it proper vascular supply, or those ones who were a little bit luckier, where it was said that they suffered from a minor heart attack. It really refers just to a minor part of the heart tissue being affected by ischemia, and therefore it is not so seriously affecting the functionality of the heart. So after completing these initial considerations about skeletal or striped muscle fibers or muscle tissue, cardiac muscle fibers or muscle tissue, it is time for us to address the third and last type of muscle tissue that we have, which is going to be very interesting because it is something without which our life would have to be very seriously altered from norm, from standard as we recognize it. This type of muscle tissue is called a smooth muscle fiber, obviously lacking stripes or striations that we have seen on both cardiac as well as skeletal muscles. I'm just adding them here to give you a little bit of a recollection of what would they look like. The lack of stripes actually doesn't come from a lack of contractile protein, quite the opposite. They are present within the cell, but this time their arrangement is not so particular. Those contractile proteins could run side to side, but could run also any length, direction. They could run front to back. And of course, as such, as they do not represent this kind of predictable pattern, when smooth muscle cell is observed under a light microscope, there isn't much of a light refraction. So we get under impression that the surface of these cells actually is showing as perfectly smooth. These cells tend to be much shorter, but this time the contractile force is not necessarily directed in one direction, for example, following the longest axis of the muscle. That would be only one of a possible direction that this muscle will experience shrinking. It could also shrink side to side, as well as shrinking in front to back direction. And this is where I dare to say that these muscles effectively could be a little bit better form of a tissue than the cardiac or skeletal or striped muscle fibers. Why do I say this? The metabolism and metabolic requirements of these muscles is a little bit different compared to cardiac and skeletal muscle. Therefore, their contraction could last significantly longer than a contraction of any of the skeletal muscles. I'll give you just one example. If, for example, you need to hold something in your hands like this, I hope you can see the small picture in picture. Let's say you're holding a couple of textbooks and you have to hold them in your hands like this. For a first minute or two, it's going to be business as usual. Third minute, you would start already experiencing a little bit of a fatigue, because in order to hold your arm in this position, you have to apply flexion at the elbow. You have to also use different muscles of your hand in order to support the book. Maybe 4 or 5 minutes into that activity, you would experience that you're running out of power, and over the next maybe a few seconds or a few minutes, depending how well trained we are, we'll be forced to say, okay, I can't hold it motionless like this any longer, and will likely drop the load. So overall, 5, 7, 10, 15 minutes, but after that, it becomes obvious. Muscles become fatigued, tired, and we have to do something in order to at least prevent more extensive damage that could be done by this kind of uncontrolled and a very excessive use of our skeletal muscles. On the other side, smooth muscle fibers, for example, you can see they are seen within the walls of a variety of different hollow structures of the body. This time, they illustrated it as being part of the arterial wall, blood vessels that are taking blood away from the body. Of course, with the contraction of these smooth muscle fibers, they will reduce the size of the lumen and indirectly will push blood further away from the heart, but also indirectly may result in increase in blood pressure in between 2 consecutive heartbeats. They're going to be seen also within the respiratory tract. Part of our airways is practically built by smooth muscle fibers that are internally lined with some form of epithelium, which is typical for that part of airways. Or we're going to see them as building the walls of our digestive tube, or maybe the urinary bladder, which is bringing us to probably the best example where I can explain the longevity of contraction that is applied to smooth muscle fibers compared to what we just said, skeletal muscle would be unable to do the same thing for a period of time exceeding 10 minutes. Let's say the urinary bladder, smooth muscles contract and they prevent involuntary leak of urine. Only when we reach that kind of critical volume of urine, basically we trigger the urination reflex and we would go to the bathroom. But if the bathroom is not available, or we're attending some kind of very important meeting, or we're in the middle of the street, we know that it is not going to result in some kind of disaster that we would have to urinate spontaneously. Quite the opposite. Smooth muscle fibers that are controlling the neck of the bladder will increase their contraction and give you needed 5 minutes, 10 minutes, half an hour, sometimes even a couple of extra hours until you finally have the opportunistic moment to visit the bathroom and to empty the bladder. So this kind of comparison very clearly points out that when it comes to longevity of contraction, it is quite clearly in favor of smooth muscle fibers. How do we control them? Do we control them voluntarily or involuntarily? Their control is also involuntary under autonomic nervous system, which, depending which part of the body we're looking at, would trigger a different effects. Sometimes it will contract or stimulate contraction, but sometimes the autonomic nervous system will have to do quite the opposite and to induce full and complete relaxation. Smooth muscle cells or smooth muscle fibers do have just one nucleus per cell, and fortunately they do have ability to increase their numbers, so we do not experience some kind of huge deficit should we have part of the body or part of the organ built by smooth muscle fibers experiencing some difficulties that eventually will result in a complete loss of muscle tissue. That is not going to happen. So contraction, of course, will be somehow mediated through the nervous system, but as we're now completing muscle part, muscle tissue, I would prefer that we do not go right away into introduction of nervous system. Let's leave it as independent and separate presentation. For now, we're going to just shrink this and we'll take a look at a few more slides before we complete the presentation of muscle tissue. So from what we were talking about over the last few minutes, skeletal muscle, cardiac muscle, smooth muscle, it is time now to make a little bit closer to what's happening in those muscles that are, simply to say, epithomes of what muscle tissue is for most people who are not necessarily receiving education for different health-related studies. When we tell to them muscles, they know, oh, it's my biceps, it's my triceps, it's my quadriceps or pectoralis major, or any other muscle that is quite common knowledge of a variety of different people from different professions. So we have here cross-section through a single skeletal muscle, not muscle cell, muscle fiber, but rather the entire muscle as biceps. And we need to do it in order to understand better what kind of composition skeletal muscles would do for all of the skeletal muscles part of our locomotor system. So we can see here that the cross-section actually is illustrating the entire muscle. Then one fragment of the muscle appears to be like telescopically pulled out. And from that fragment, another smaller item is further pulled out. And if we see the hierarchy, essentially we're talking about single muscle cell or muscle fiber, which again by itself is ineffective. Multiple muscle cells or muscle fibers will be held together in order to form the muscle bundle or fascicle, and then multiple muscular fascicles are required to put them together until we finally observe formation of a full quality muscle such as biceps, triceps, latissimus dorsi, or any other muscle that comes to your mind. Individually, muscle cells, fascicles, and muscle in general are enveloped with more connective tissue. So in this case, we're talking about endomysium, connective tissue surrounding individual muscle cell, perimysium, connective tissue that envelopes the entire fascicle, and then we have epimysium, connective tissue that will be required to cover and to surround the entire muscle. So endomysium inside, perimysium for fascicles, and epimysium for the entire muscle. Because these connective tissues practically expand past the most distant boundary of the muscle cell or muscle fibers. Basically, at the end of a muscle, endo, peri, and epimysium come together, and that kind of coming together, they assemble a tendon of a muscle. Practically, we can say the intermediary byproduct, which ensures that muscle does have means or tools to attach itself, to insert itself onto the surface of a bone. Of course, a muscle has to do it at least twice and at least across a single joint. Why? Because there is no benefit if a muscle would be attached with its two ends onto a single bone. That bone by itself doesn't make any joints. So contraction of a muscle, let's say being attached twice on my forearm bones, would be absolutely ineffective. There is no need to keep bending or trying to bend bones of the forearm. So muscle with one end will have to attach somewhere on my arm, and with its other end, it will have to be attached anywhere on my forearm and it will cross the elbow joint. So contractions of the muscle may lead to elbow joint flexion, or if it's on the other side of my upper limb, it will be involved in extension of my elbow. Basically, tendons are named as tendons of origin and tendons of insertion. Or we can just say common denominators. Our tendons are used as the attachment points for a muscle on a skeleton. When we use the words such as tendon of origin versus tendon of insertion on the same muscle, basically it is meant by tendon of origin is on a stationary part of the body, whereas the tendon of insertion is on a part of the body which performs the movement when this muscle becomes activated and forced to do contraction. So if we're looking for a muscle passing between arm and the forearm, arm technically becomes origin, forearm will be seen as insertion if the muscle is creating flexion at the elbow, forcing my forearm to come closer to arm, stationary part and movable part, origin, insertion. All right, we don't have to worry so much about this. I just want to prepare you for things that will come later on. And that's why we are benefiting from a little bit of extra explanation. What is origin? What is insertion of a muscle? Let's now take a look at the lower part of the screen where this single muscle fiber is illustrated again, but this time with a greater magnification. And when we cut open its membrane, its cellular membrane, we're going to see inside of the cell, those dark spots that you see more peripherally, of course, are multiple nuclei, and I still owe you explanation of why muscles have multiple nuclei. We'll do it shortly. However, you can see here that inside a single muscle, we would have plenty of additional cylindrical structures that run the length of a muscle, and those cylinders, we call them myofibrils. And at the surface of each and every myofibril, we see some other lines that are kind of intersecting each other and overlapping each other. These are striations, and that is essentially why do we call this skeletal striated muscle tissue or striped muscle tissue. Each of the myofibril would be built by hundreds of protein fibers overlapping each other, generating these alternating light and dark bands. So that is the contractile force within the muscle. Individually, we call them myofibrils, and we're going to break down the myofibril furthermore until we reach a level which is easy to explain for the physiology of muscle. And of course, that's going to be a sarcomere. Word sarx, S-A-R-X in Greek, means flesh or meat. So that's why we're talking about sarcoplasm or sarcolemma. Sarcolemma is a cellular membrane, cellular envelope, but specifically related just to muscle tissue. And sarcoplasm would be the inside of a muscle fiber. Otherwise, we would call it the cytoplasm. Still, we need to explain the structure of a myofibril in one single subunit that would be found within it. But before it becomes completely missed, let's immediately add here interaction between nervous and the muscle tissue. Specifically, skeletal muscle fibers. They cannot contract on their own. They need a stimulus. And of course, that stimulus doesn't come from any other part of the body rather than our nervous system and the peripheral nerves that will be very intimately related to muscle fibers or to muscles in general. So the very end part of the nerve cell, which we call the axon with its collateral branches, practically reaches its end. And this end is what is illustrated on this photograph. That is axon terminal. Axon is part of the nerve cell, and this is its terminal, axon terminal. At the axon terminal, tiny little enlargements are observed, and they're called the synaptic end bulbs. This is where nerve through synaptic end bulbs practically comes very close onto the surface of a muscle. And then it would trigger a series of changes within a muscle that will lead into muscular contraction. So nerve doesn't plug itself directly and passes its electrical current into the muscle. Quite the opposite. Nerve carries its own electrical current along the axon, and when the current reaches axon terminal, basically it triggers a bit of chemical reaction. Here at the end, this is called the neuromuscular plate, this area of interaction between axon terminal and this part of the cellular membrane, that will lead to a number of different changes inside the muscle, forcing contractile proteins to start interacting with each other differently, which is actually muscular contraction. On the next slide, we're going to see a diagram of myofibril, and we're going to see here what happened in our past. During embryonic development, we have been observing a variety of different young productive cells. All of them would have a suffix blast. So, for example, if a cell is called chondroblast, it forms chondros, which is cartilage. If a cell would be called osteoblast, its responsibility would be to produce bony matrix that later would calcify, turning it into bone. So we have myoblast cells, obviously young productive cells that multiply relatively fast in order to increase the number of units, which is required to populate the entire developing embryonic or later fetal body. But something unusual happens between myoblasts. Their membranes fuse with each other, and multiple cells are arranged into structure that we normally call syncytium. So syncytium is really like having no specific prescription how to make it. It is enough to think of it that syncytium or syncytial cell has been made by blending of multiple structures, multiple cells into one single structure. And in this case, fusion of myoblasts are elongating this skeletal muscle fiber, making it longer, but each and every cell that comes and blends into it essentially is adding one more nucleus, which is the reason for we see multiple nuclei within muscle fibers of the skeletal system or striped muscular fibers. At this point, I would not prefer that we go directly into physiology of muscle tissue and muscular contraction. That will be followed up by multiple slides that are coming after this one. I prefer to talk about sliding filament theory as a separate presentation. So if this part that we just completed doesn't really bother you because you have a good grasp, good information about it, you can move on directly to a video that will be titled Sliding Filament Theory, and you can observe or learn things that are a little bit more complicated or something that you still need to cover in a course that you're taking. So this would be the third and penultimate part of tissue videos. Next one that will be posted will be about nervous tissue, and that one will also be more of informative nature as what do we expect within the nervous system, but physiology of nervous tissue will be explained in a separate video that will be likely titled Action Potential. I hope that you enjoyed this presentation and that I will see you shortly I'm inviting you to leave some kind of comments if you like what I'm doing. I'm also inviting you, if you really like it, to spread the good news between your classmates. Point out to my YouTube channel. It has been titled Anatomy Made Simple, because when I started it, my focus was really on the anatomy, mostly of the skeletal system, because that's what I had to do at that time. But I realized that it's not only about anatomy, physiology is also a very interesting, a little bit more lively part of the human body. And if I could do something to make your lives a little bit easier and a little bit less challenging for perhaps upcoming exams or colloquia or assignments, well, that is why I'm doing this. So hope to see you soon, and everything best in your studies. Take care and stay tuned for more videos to be posted on this channel. Thank you very much for watching this one.